Hello everyone and welcome to Instant Biology by Dr. Neelab. The topic that we have to discuss today is optical properties of amino acids. Now this is a lecture in the lecture series of Basics of Biochemistry. I have already made a video in Hindi on this particular topic. So if you are comfortable in Hindi language, then you can switch over to that video. Otherwise, this video will also comprise of all the important things in this particular topic. So let us start. We would try to establish a link between the chirality and the optical properties of amino acids. So everything that we would be talking about in today's lecture would be with respect to amino acids. So we would consider chirality and we would also try to establish a link between optical properties. Okay, so the first thing that I want to make sure that you understand chirality. What is chirality? Chirality is the condition when all the four valencies of a carbon are attached to by different elements or atoms. For example, if you see on your screen, you can see this structure. This is a carbon, and attached to this carbon are different elements A, B, C and D. You can see over here A, B, C and D. So these are the different elements that are attached to carbon. Now this kind of arrangement, when there is no repetition of a particular atom or there is no double bond present in the carbon, then this type of arrangement or this type of condition can be considered as chirality, or this particular carbon to which the different elements are attached would be considered as a chiral carbon, or you can also consider it as no center of symmetry. That means this carbon has no center of symmetry. It is not symmetrical. It is not providing any symmetry. Okay. So this carbon can also be considered as a chiral center, or you can also call it as chiral carbon. If you are considering in terms of symmetry, that is whether the molecule or whether the compound that you are talking about is symmetrical or not, then you would say, since there is no center of symmetry, since the carbon that is present in the center is chiral, no center of symmetry can be seen. So this is one condition which you require for chirality. That means there should be no center of symmetry, and the carbon to which the different elements are attached, all the elements should be different. Or in other words, you can say there should be a chiral center or chiral carbon that should be present. Now, when we are talking about large molecules, then what happens is we should also consider a concept of plane of symmetry. So what is a plane of symmetry? Plane of symmetry is just a plane that can divide the molecule into two equal halves or two symmetrical parts. For example, if you see in this second structure, this is a carbon. It has been attached by A, B, C. Now this carbon is also attached to another carbon that is again connected with A, B and C. Now this plane that can be used over here, if you divide or if you break by this plane, if you break this molecule through this plane, what will happen? Two identical or two symmetrical structures would be present. Now this is called as plane of symmetry. Now if you are referring to a particular molecule as having plane of symmetry, then it would not be considered as a chiral compound or chiral carbon. Okay, so if there is a plane of symmetry that can divide the molecule into two equal halves, then in this case you would not consider it as a chiral compound. Okay, that means there are two conditions: that there should not be plane of symmetry and there should be no center of symmetry if you want a compound or a carbon to be chiral. Okay, so what are the important parameters or what are the important things that you should consider if you are referring to something as chiral? So if a molecule is chiral, then there should be no plane of symmetry as well as no center of symmetry. I believe this would be clear to you. So when both of these are not present, then you consider that molecule as chiral molecule, and if both of these things or any of these things is present, you would refer to that compound as achiral, or you would refer to that carbon as achiral carbon. Now let me come back to the concept of amino acids. In case of amino acids, you would know that there are 22 standard amino acids, and in those amino acids, the most simplest amongst them is glycine. Why is glycine the most simplest? Because the R group that is present in glycine is having a hydrogen. Okay, so you would be knowing about the structure of amino acids. If not, I'm just going to revise it. So in case of an amino acid, there is an carbon. Attached to carbon can be an amino group and can be an carboxyl group and also a hydrogen. One extra thing would be an R group, right? So this should be an R group. But over here you can see that the R group is also hydrogen. That means the condition that we had just studied, where we said that on a particular carbon all the elements that are attached should be different, right? So that condition has been broken, or that condition is not fulfilled over here. So if the condition is not fulfilled, you will consider this compound or this carbon as achiral. That is why we say that glycine is achiral, it is not chiral. Okay, so it is the most simplest of amino acids, and it is also achiral in nature, because this hydrogen as well as this hydrogen are same. That is why you are considering it as achiral. So this was the first letter, this was the first mention in the slide, that all amino acids except glycine are optically active. Now we would link chirality with optical activity in just a couple of minutes, but I believe you would have understood that glycine is achiral. Next, when a compound is chiral, then it will definitely show some optical properties. Now what do I mean by optical properties? I would just tell you in a bit. So again, remember that if the compound or if the molecule is chiral, then it will definitely show some kind of optical activities. We would talk about what optical activities are actually seen over here. So what is optical activity and how is it measured? Optical activity is basically the ability of an optically active compound to rotate the plane of linearly polarized light. Okay, so I would tell you what linearly polarized light is, but for now just consider it as a form of light. When this light is passed through let us say a solution of amino acids, and if this light tends to get rotated from its main axis, it tends to get rotated either clockwise or tends to get rotated in anti-clockwise direction, then this kind of solution or this kind of amino acid would be considered as optically active. Again, I'm trying to remind you that optical activity will only be there if the compound is chiral. Okay, now I've told you that what optical activity is. That means the plane polarized light would be rotated either towards clockwise or anti-clockwise. But if I want to quantify this, how much is the angle of rotation? How would you quantify this? So we have a very good parameter for that. That is called as optical rotation. You can actually measure that what is the quantity of the rotation of the light, or what is the angle of the rotation of the light. This is called as optical rotation. So as I've already said, optical rotation, in case of optical rotation, you would get the quantitative measure of the rotation of light caused by the compound. Okay, quantitative measure, how much light or how much rotation has been there, you can get the value of the amount of rotation through this. Okay, so this I believe would be clear to you. Now coming to what are the factors on which the optical rotation would depend. The first thing is, it depends upon the concentration of the compound. Now it is very easy, it is very logical also, that if I use a larger concentration of an amino acid solution, then definitely there would be a change in the angle of rotation. The angle of rotation can be more or less depending upon the concentration of the amino acid that you've used. This is the first point. The second point is temperature. Now understand this, that rotation depends upon the temperature that you are doing the experiment on, and it also depends upon the lambda. Lambda is the wavelength of the light that you are using. Okay, so this I think clears the second point, and as well as the third point, because the third point states that the wavelength of the light used. The fourth point is solvent that you are using. Now you would definitely be dissolving your amino acid in one particular solvent. For example, if you are using the universal solvent, that is water, or let us say you are using some other kind of a solvent, some organic solvent, then using different kinds of solvent may change the optical rotation. Coming to the final point, the final point over here is light path length. Now what does light path length means? Now suppose this is the cuvette in which you have kept your amino acid solution. Now this cuvette has some kind of a diameter. Let us say this is the path length, or this is the this distance is the distance through which the light has to pass in the amino acid. Now if I increase the path length to this much, or if I decrease the path length to this much, now you would clearly understand this, that increasing or decreasing the path length will definitely change the optical rotation, because light has to travel more in the amino acid. If you are increasing the path length, light is traveling less in the amino acid if you are decreasing the path length. So again, optical rotation would be clear to you, and the factors that on which the optical rotation depends would be clear to you. Let us move on to the next point. Now there is a mathematical expression that you can use for solving numericals when it comes with to respect of optical rotation. Now what is this formula? Now you can see that A is equals to alpha T lambda, you can see over here T, you can see over here lambda, into C into L. Now what is this? This is observed rotation in degrees. That means what is the amount of rotation that is taking place, and you have to measure that in degrees. What is C? C is the concentration of the solution in gram per ml. Now remember that the concentration should be present in gram per ml only. What is L? L is the light path, and that should be in decimeter, because it is the unit of distance, right? So it should be present in decimeter. Okay, and what is this alpha T lambda? Now understand this, alpha T lambda is specific rotation of the compound. Now this specific rotation should be constant at a given temperature and wavelength. Now if you change the wavelength and temperature, definitely this alpha would change. So alpha is specific rotation, A is observed rotation, C is concentration, and L is light path length. Now you should remember the units that you have to use with this. Let us consider an example, and through this example we would understand that how we can do this experiment, or how you can analyze, or how you can tell that what is the angle of rotation, how much is the light rotating, or whether the compound is optically active or not. We will consider all this. Now if we are performing an experiment, and in this experiment we have a light source. Now this light source is a sodium lamp. Now you would understand this, that sodium lamp would create certain, so this sodium lamp would have light, or would have wavelength of light that would have different directions. That means it would have different wavelengths, as well as it would have the rotation in different directions. Okay, so sodium lamp is providing such light that is having different wavelengths and different direction of rotation. Now you can see that although wavelength would be also be different, and this direction of rotation can be like this, 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 anything it can be possible. Now this particular light from the sodium lamp is allowed to pass through monochromator filter. Now what is monochromator? As the name suggests, monochromator simply means that this will allow only one wavelength of light to pass through it, and all the other wavelength would be stopped. That means the light that is passing from here would be just having one wavelength. It will not have other wavelengths. So just one wavelength would be present here, that is passing from the monochromator filter. All the other wavelengths would be stopped by the monochromator filter. Now this one wavelength, this will have multiple directions. This you need to remember. So you can see this is the direction, 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 this is the direction. But means all the directions are possible in this particular wavelength of light. Now when this wavelength of light is passed again through a polarizing filter, what will happen? All the other directions would be cancelled and only one direction, that is up and down, would be favored. All the other directions would be cancelled. So you can see that after passing through the polarizing filter, you can see only one direction is present like this. I believe this would be clear. Now this particular light is called as linearly polarized light. This you will call as linearly polarized light, LPL. Now this linearly polarized light is again passed through the sample. Now what is your sample? You can use, for instance, amino acid solution. Now if the amino acid solution is glycine, it will not rotate the plane polarized light. I believe you would have understood this, because it is not chiral. It is achiral. If it is achiral, it will not have any optical rotation properties. But apart from glycine, if you use any other amino acid, then what will happen? There are chances that you can see some optical rotation. How is that possible? Now suppose this was the direction of the plane polarized light. Okay, this was the direction. Now when you are passing through the sample cell, or when you are passing through the sample, there are chances that the light is rotated towards the clockwise direction, as you can see in this diagram. So you can see that the light has been rotated in the clockwise direction. In the red you can see the new direction of the light is now like this. That means the rotation is in clockwise direction like this. So when the rotation is in clockwise direction, you can understand this, that the solution is dextrorotatory. Dextrorotatory is abbreviated as small d, and the direction clockwise is shown by positive symbol. Okay, so whenever there is clockwise rotation, it is called as dextrorotatory. The compound is called as in D form, small d form. Remember, capital D and small d are different. Over here we are talking about small d form. Small d represents the dextro form. Okay, so or you can call it as the positive form. If I talk about some other kind of an amino acid, now it can rotate the plane polarized light towards the left hand side like this. In the second case, you can see over here. Now over here the light has rotated towards the anti-clockwise side. Anti-clockwise is called as levorotatory, and this levorotatory is shown by the letter small l, not capital L, small l. Now this small l, this anti-clockwise direction can also be represented by a minus sign. Okay, so now what we have done is, we have, on the basis of optical rotation, we have divided or differentiated the two types of amino acid solutions that can be formed. One can be D form, rotating the plane polarized light towards the clockwise direction. Another one can be the L form, rotating the plane polarized light towards the left direction. Now one thing was left. In case of alanine, for example, in contrast to glycine, if I consider the concept of alanine. Now this alanine has an alpha carbon, all right, carboxyl group, amino group, H group, and in the R group there is a CH3 or methyl group. In the place of R group there is a methyl group. Now because of the presence of methyl group, remember glycine was the simplest of amino acids, it was having hydrogen in place of R group, but over here this is the second most simplest amino acid. This is alanine, and in place of the R group it is having methyl group. So over here, because of the presence of methyl groups, all valencies are different. If all valencies would be different, it would be having a chiral center or asymmetrical center. And because of this chiral or asymmetrical center, what will happen? This would show some optical rotation. Okay, so this will definitely show, alanine would definitely show some kind of optical rotation. Now I believe you would have understood this. I would also be talking about certain numericals now that can be asked in different life science examinations, and these numericals would be based on the concept that we have already studied. So for example, there is this question that the positive enantiomer of compound A has an optical rotation of 125 degree. If pure sample of A has an optical rotation of 100, what is the composition of the sample? Okay, now this positive enantiomer's rotation has been given to you. Positive enantiomer's rotation has been given to you, that is 125 degree. Now using this concept only, if positive enantiomer's rotation is positive 125, then the negative one would be negative 125. So negative enantiomer would have a rotation of minus 125. The pure sample or the pure compound A is having the rotation of 100 degree. It is having the rotation of 100 degree. Now let us, what is being asked to us, that what is the composition of the sample? That means how much amount of positive enantiomer and how much amount of negative enantiomer is present. So now let us consider, let us assume the total amount of sample B X, or let us consider it as 1 for our ease. So let us consider that the total amount of the sample be 1. Now let us consider that the positive enantiomer is, the amount of the positive enantiomer is X. So positive enantiomer is X. Let us consider that this positive enantiomer is X. Now what would be the concentration of the negative enantiomer? It would be 1 minus X, because positive was X, total amount was 1, so this would be 1 minus X. Okay, so you know that the rotation of the positive enantiomer is 125. So let us consider this X into 125, and we also have to consider the rotation by the negative enantiomer plus 1 minus X. This is the concentration or the amount of the negative enantiomer, and this is the rotation that it will lead to, minus 125, and this would be equal to 100 degree, because the final rotation that can be seen is 100 degree. Now let us solve this. 125x plus 125x minus 125 is equals to 100. Now calculate this. 250x is equals to 225. X is equals to 225 upon 250. Now if you want to calculate the percentage, multiply it by 100. Now X would come over here to be around 90%. That means the positive enantiomer had a concentration of 90%. Whereas if I talk about the negative enantiomer, it would be 1 minus X. This would be 100, or let us say 100 minus 90, this would be 10%. Okay, so I believe this would be clear to you, that the positive enantiomer would be 90%, negative enantiomer would be 10%. Okay, this was the concept. Let us talk about another question, and in this question, the question says a pure sample of naturally occurring chiral compound A 0.50 gram is dissolved in water. So you would be knowing that this is the concentration of the compound that has been given, and the solution or the path length has been given as 0.5 decimeter per cent. Plus there are 3 polarimeter readings that have been recorded, this reading, this reading and this reading. They are asking you what is alpha. Now you know from this formula, A is equals to alpha T lambda into L into C. You already know this formula. And since you have been given that the 3 polarimeter readings, that means 0.625, 0.706, 0.682, 3 readings have been given, so that you can find out the average amongst these readings. So let us just calculate the average. What is the average? If you calculate this, then 0.625 plus 0.706 plus 0.682, you divide it by 3. This would give you the average reading. Now, if you divide it by 3, then the value that you would be getting would be 0.671. This is the average value. That means this is the A value. Okay. Now, what is alpha? We have to calculate it, and we have been given the concentration as well as the length. Now, let us put this everything in the formula. So, A, that means 0.671 is equals to alpha into C into L. So L, we already know it is 0.5, and C also we know that it should be 0.050 divided by 2.0. Okay, because it is in gram per ml, so you can directly use this in the formula. Now what you would get is, if you solve this, alpha would come out to be 53.68 degree. So this would be the answer of this question. So these type of questions have already been asked in optical rotation concept. I hope your concept about this would be clear. If you like this video, please hit the like button, subscribe to my channel, and also share this video amongst your friends so that all of you can get benefited. Thank you so much and have a good day.